Hello, and welcome to this podcast with Cyano Biological. My name is Will Suter. Today, we'll be talking with Dr. Amy Sheng, a technical account manager at Cyano Biological, and Dr. Lurong Pan, the founder and CEO of Ionosensor, an AI-powered next-generation biotech startup. We'll be discussing how AI computing can be used in combination with high-throughput production and screening of proteins to expedite the development process for candidate universal vaccines, an ideal method for preventing and controlling future pandemics, but also one of the major research challenges in biotechnology today. So thank you both for joining me today.
Thank you for having us.
Yeah, thank you. It's my pleasure.
So Dr. Sheng, let's start with you. Can you tell us a bit about your background and your current role at Cyano Biological?
Thank you, Will. I'm a technical account manager in Cyano Biological. My background is cell and molecular biology, antibody development and production. Currently, I'm in charge of the CRO program in Cyano Biological US.
Excellent. And Dr. Pan, same for you. Can you tell us a bit about what you do and your background and what brought you to Ionosensor?
Yeah, thank you very much. Yeah, I'm the founder and the CEO of Ionosensor, a AI drug discovery platform. I have a background in computational biology as a PhD, and also a background in computer science master and specializing in artificial intelligence. And I have been in this industry doing drug design, leverage different computational algorithms for about 14 years. And then I was initially collaborate with Cyano Biological, using them as one of the cell providers of our internal drug discovery program. And then we happened to find some common ground to collaboratively break up this technology barrier using like a combined computational and wet lab strategy. That's why we're here as a team.
So to both of you, really, can you tell us a bit about universal vaccines? How would you define it, and why is it such an important thing to work on?
In general, by definition, universal vaccines are vaccines that provide the broad efficacy against various strains of a virus. From the case of SARS-CoV-2, we have experienced and understood how rapidly a virus can mutate and can escape the immunity. And therefore, it is particularly important to be able to develop the vaccine or therapeutic reagents that will continue to provide a protection against the new versions of the virus that may emerge. A universal vaccine has more potential compared to the traditional ones to protect vulnerable populations from various strains and even future variants. So before SARS, COVID, influenza has been a target to develop a universal vaccine. And in fact, the development of the influenza vaccines with broader protections has been a goal for really decades. However, in recent years, due to the new vaccine targets and more efficient delivery platforms, it is believed that this goal will become more achievable.
What are some of the other advantages that universal vaccines have over traditional methods of vaccination, doing sort of a disease at a time?
Yes. So let's take the influenza again as for example. The traditional vaccines do not produce durable protective immunity and a cross-reactive immune response that can neutralize a diverse influenza virus strains. Their efficacy varies significantly in various age groups and against the different viral variants. And indeed, the strain mismatch is one of the main causes of vaccine failure. And very commonly, you hear friends saying that, flu shots do not work, and I was knocked down for one week by the flu, even though I got the flu shots. It's not the flu shot; it's actually just a strain mismatch. And in fact, just last year, the major strain of flu H3N2 mutated so significantly in a way that the designed vaccine could not efficiently provide immunity to the population at all. So, however, universal vaccines could potentially solve this issue and provide better protection as quickly as possible. And besides the efficacy, universal vaccines would lower the cost of the vaccine R&D, manufacture, and stockpiling if we are faced with a rapidly mutating virus, which will greatly benefit the low-source country populations that are particularly vulnerable to the pandemics.
So can you break down some of the science behind how universal vaccines work? How are they able to provide immunity against a range of diseases?
Universal vaccine development mainly focus on the unchanged part of the virus, or what we call conserved regions. However, this region might be shadowed by the ever-mutating domain of the virus, which we call immunodominance. In other words, the generated universal vaccine might have lower immune responses. So now researchers try to make a chimeric protein vaccines to make the conserved region more immunodominant and the immune response broader and more durable. How the vaccine packed also greatly affects the strength and the quality of immune responses. For example, the adenoviruses, the naturally infected human ones that can be modified and used to deliver the DNA sequences encoding the viral antigens we want to present. So the good thing about this is that they can keep producing the antigen for weeks, which might help extend the response. There was one study in Mount Sinai working on the H1N1 virus. They generated an adenoviral vaccine and tested it in mouse. And besides the H1N1 strain, and they exposed the mice to other strains that are distantly related. And they found that for those mice, the survival rate is significantly higher than those who only received the traditional H1N1 vaccine. Therefore, showing that how the vaccine packed and delivered are also important in the universal vaccine design.
I have a few points to add in terms of the three aspects. So for the needs of it, definitely, I think it is quite urgent. As you can see, the pandemic. The vaccine has two types, I would say. One is preventive, another is like therapeutic. So preventive means you give it ahead of time and it's protective for a while. And those normally the vaccines that generates B-cell immunity that has longer memory in our immune system and that take effect. And in this case, from I think the technology feasibility and current improvement in science, I think we are already observe certain antibody or antibody cocktails that are able to target multiple like viral infections, no matter it's the same family or even cross family. So I definitely believe if we would be able to find a universal antigen using both computational and experiment measures, there's definitely achievable of doing a universal preventive vaccine that generates a cocktail of long-term like protective B-cell immunity. And another kind is therapeutic vaccine. I think it's also like reasonable in terms of science. For example, another part of our immunity is T-cell immunity, which normally give probably a more instinct and a more of a harsh response to kind of clear out a lot of those non-selective invaders of our body. In that case, I think if we could really control T-cell behavior in a therapeutic way that stimulates its function when we get infected, but at the same time to customize for different type of patients. Some patients we know they have compromised T-cell immunity, and some patient has like a overexcited T-cell immunity that caused more of a inflammatory effect, so that if we could somehow have a type of a modulator to stimulate our T cell to be able to protect us at the same time, not hurt us, but clear out those any type of invaders, pathogenic molecules from outside, that it will be also be some kind of universal vaccine. But it's just being used for after you get infected, that we just try to stimulate your immune system. So I think both technology are currently I think probably 10 years ago, people won't be able to imagine it. But I think now it's come to the time that we realize that it's achievable to a certain extent because we know more about those functions and we explicitly studied their function and sequence and also in the cell biology base. So, and it can be quickly compute and quickly validate in the wet lab so that a lot of those hypotheses or large scale of computation that couldn't be done in the past are achievable now. So that I think as long as we really fully understand the B cell and T cell function of our immune system, we'll be able to design pretty good universal vaccine in the future. And another thing is from technology perspective is the modality of vaccine. Like Amy mentioned earlier, we can have protein as a form of a modality to mimic a universal antigen. Or we could have like a mRNA vaccine and or DNA vaccine or all type of modality that could potentially generate a universal protective B cell or T cell response that would be also available. And at the same time, we'll probably even be able to make a universal vaccine as a oral available drug as well, if certain delivery strategy is good enough, not just for injection. So in that case, it will be more available to most of the countries that has no medical facility to give injection. So I think that's also a direction that this industry can explore. Because as far as we know that we already can deliver peptides as an oral available form, which is definitely already a small sequence of a protein, which is also be able to be like an antigen of immunogen. So that it's possible even in the future we'll be able to have a orally bioavailable universal vaccine. That would be good.
So what are some of the challenges in finding candidates for universal vaccines, and how can AI technology help to address those challenges?
That's a very good question. I think first of all is in the past, it's like we have to generate, for example, antibodies using a single antigen species. For example, the COVID, initially we designed it or we kind of using animal model to generate it as a strain that was currently happening. But when it's mutant, we have to basically give, if it's an animal as the experiment subject, we have to inject it multiple times in multiple timelines. So which is really, we can't really chase the speed of the mutant to do the wildlife experiment using conventional animal model way to develop that. So in that case, I think computational technology could be a great help. Such as we have a collaborative experiment with Cyano, we have virtually computed all the strains from the beginning of the pandemic to that current time point over 1,000 different strains. And just within a few hours, you can generate those antibodies that potentially target different strains. And on the opposite, we can also using the same algorithm to identify what is common among all those mutant strains so that we would know if we can generate antibody to prevent the future strain. And interestingly, that result shows that using this model to design the antibody, we can also, this antibodies also be able to prevent a strain comes later, which we surprised as well in our lab. So that it must be some patterns in those evolutionary traits of those species that we can learn in the past and to be able to predict the future mutation trend of those species. And if we can completely digitalize those virus, and we think we could definitely using some type of algorithm to find its evolutionary pattern and be able to find the common motif or common immunogenetic sequence to contribute to that process. I think definitely computational technology could speed up that process and even discover new phenomena in biology in the future, if not currently already happening.
So, Dr. Sheng, from your point of view, how has the introduction of AI and working with companies like Ionosensor, how has that helped and changed your sort of workflow when you're working on sort of developing technologies like these?
Yes, so AI Innocence is definitely a great platform for to design proteins and design antibodies and provide us a guideline of what will be the best ideal candidate looks like. Instead of like searching in the dark, AI Innocence by the computational ways, we can have a better understanding and also a prediction of how the coming out products will interact with the protein partners. For the challenging of the developing universe, so I think Dr. Pan already pretty exhausted the different kind of aspects of the questions. Would like to add a little bit on the production side. So if the vaccine is in the protein form, so the production of the predicted sequences can be sometimes challenging too. The desired protein should be with high stability, high yield, and purity for the later manufacture and other aspects of the R&D. So we will have to perform the high throughput production and screening to find the ideal candidates. So it's not always as straightforward. We have to do a lot of troubleshooting to standardize the production of that protein.
Can you talk a little bit about how you started working with Ionosensor and how you developed the AI platform and integrated it with your development workflows to work on these new vaccine candidates?
Yes, so Ionosensor was one of our customers. We developed a lot of recombinant antibodies projects together. Since we provide various platforms for recombinant production, especially the high throughput protein production and screening, we're able to produce up to 1,000 constructs in turnaround time as short as 4 weeks. So together with AI Innocence, we're able to provide quick answers to some basic information such as the ones I mentioned, the yield, stability, and purity, and regarding the computational designs. Also, we have extensive experience with viral protein productions, and as we mentioned, we have the largest viral protein bank in the world, and the product line covers the hemoglobin, HA, NP proteins. So from all WHO recommended vaccine strains in recent years. So these antigens can be used to analyze vaccine-induced antibody response.
And how about you, Dr. Pan? Can you talk about the process of sort of adapting to your technology to what Cyano needed in this project? And how does working with Cyano compare to other projects that you have?
Yeah, so we started with the collaboration on the COVID project, like Amy was mentioned, because they are really has good expertise in this field in terms of production and also some kind of experiment measurements of those antibodies properties. And then we later find out that a lot of the customers also not just wanted a production of a protein drug, they also would like to improve the property of those protein drugs. And in the past, in the industry, you have to kind of blindly, kind of in a random space to try different mutation to find a good quality one. That would take a lot of time and also cost of experiment. But the most thing is the time. So in that case, we find out a good niche that if AI could involve and be able to shortlist all those unnecessary experiments, because most of the random generated mutation are negative in the real experiment test, so that we can enrich all the high probability positive species using our AI engine and only do very limited small amount of experiment. It would definitely save time by at least, I think, 50 to 60%, if not 80 to 90%. So in that case, it would expedite the R&D process. And at the same time, from the customer side, they would also spend less money to have those failed experiment paid instead of, just get the good result and be happy. So in that case, it's more of a good solution for the industry. And at the same time, if we combine to have a design and a production capability together, we can really have a quick turnaround for more customer and more deals being made. So we'll find out it's a good business model for both of us as well.
So looking to the future then, I guess the universal vaccines have been talked about as a goal for quite a long time. but obviously it's quite a big ask. It's quite a big challenge to sort of, get them into production and into actual therapeutic use. With COVID in particular, because of the huge project to develop vaccines for the pandemic, obviously the potential timescales of a lot of vaccine development has changed quite radically. It's no longer years and years. We now know it's possible, at least with focus, to do it in months. How long do you think we're looking at until universal vaccines are common in therapeutic use around the world? And how much is AI affecting that? Is that going to shorten that timescale by a couple of years, by 10 years? How much of an impact is that having?
It's a good question. So currently, actually, I think a lot of universities and commercial entities and also nonprofit organizations such as the Gates Foundation and its affiliated SEPI also has given grants to different people to do it. We also applied for it. So I think right now, like the entire world is working on it for sure. I think that's definitely a good sign. And from the scientific and technology readiness point of view, I think it's getting close. I can't really make a accurate prediction is 5 years or 10 years, but I think we would be able to see some kind of good animal study result in the coming 3 years. I would say we can validate that in animal model, but for a large trial on human, that's another story, because that would involve a long-term and a series of safety studies and also a larger population collaboration globally in different regions. So that's I think at least, even though the COVID will take a very short time to get the vaccine, but normally a vaccine could be as long as 20 years of trials. I think, let's see, it's rationally speaking, I would foresee, I think 5 to 10 years, we'll probably be able to get it validated on human trial for, let's see, not completely universal, but I think to be able to cover a majority of the common pathogens.
And what are your thoughts on that, Dr. Sheng? Do you agree?
Yes, I absolutely agree, because what we've seen recently for the SARS-CoV, it's just a very, very unique cases of the vaccine development. And it's really amazing that how at that exact timing that mRNA vaccines and other type of COVID SARS vaccines came out so rapidly, even though the efficacy towards the Omicrons dropped a little bit of those current available vaccines, but still it was able to cover quite a lot of variants of the current variants. But as Dr. Penn mentioned, the normal process of the universal vaccine development or any vaccine developments would just take a very longer time, especially on the human trial, to make sure that the vaccine have a good safety record and finally being approved by the government. So yes, we will hope to see that coming out faster, but let's just say let's keep it safe and keep it on track.
Great. Yeah, that's good. So do either of you have anything else that you'd like to add? And that's all the questions I've got.
Can I ask one question for Dr. Penn?
Mm-hmm.
Oh, sure.
Yeah. So with all different kind of viruses like influenza or SARS or other type of viruses during the vaccine development, do you see any kind of a similar evolution patterns between different kind of virus?
Yeah, that's a good question. There's been interesting study earlier. I think it's University of California, San Francisco. They have conducted a multi-omics study using primary proteomics. Basically, what they do is they compare the protein composition of the immune response of different virus and different, even like bacteria infections and some parasites even. And then they have found some shared common like antibody sequences or some other responded immune particles that share similar profile. So they're definitely not a single thing that is shared, but I think it's a pattern of a group of molecules. They should have some kind of pattern that being shared across species. And that's the fundamental, let's see, evidence or assumption in this entire universal vaccine theory, right? If there's nothing shared, we can't really do it. And it's interesting. I remember, like, for example, Earlier, there's been observation hypothesis that the TB vaccine, which has been used for 100 years, I think, could generate similar immune profile of proteins as some other viral infection, including the COVID. That's why the TB vaccine was actually one of the largest trial for both therapeutic and preventive on the COVID study, which is also kind of interesting. So I think there's some scientific ground. Different species share similar genomic pattern. And at the same time, we have 8 to 11% of our human genome is intraviral genome, which is virus that has been not existed yet, but humans somehow remembered in our genomic material. And they are expressed in certain cancer cells and other events in our body. So in that case, we are also a recorder of this evolutionary pattern as a human species to associate with viral species, which is also interesting as an interesting biology phenomenon. So I think together speaking, we definitely see common patterns for viral evolution, which they evolve pretty quickly and also along with their hosts. And I think it's something we can definitely learn digitally and computationally to either analyze understanding or at the same time even predict its trend. And that's something I definitely have belief for, because everything is encoded in an RNA or DNA string that is a shared digitalized way for all species. So in that case, it's definitely something computable. And yeah, that's all I would say. I hope that answered your question.
Yes. Thank you.
Thank you.
Excellent. All right. Well, thank you both of you. That was fascinating. Thanks for your time and sharing your expertise with us.
All right. Thank you.
Thank you, Will.